Welcome to the Chemical and Functional Genomics Lab led by Chris Gonzalez. My name is Hala Fass. I'm a senior research scientist. Our lab focuses on two main areas relevant to the environment and human health: the natural product discovery pipeline and the high-throughput drug screening. Our main project focuses on the discovery of new anthelmintics. More than 1.5 billion people are infected with helminth worm parasites, affecting primarily marginalized populations in low to middle income countries. A few drugs are available to treat those worm infections, but a rising resistance is seen among several parasites, which causes a challenge to the control efforts. Using the living free nematodes C. elegans and the distantly related Pristioncus pacificus as model animals, we screened several chemical libraries containing approved drugs, natural products, and bioactive compounds. The screen confirmed the effects of non-anthelmintic on these worm models, thus validating our approach. Among these, we identified a group of natural compounds that kill C. elegans at all developmental stages and cause mortality in the veterinary parasitic nematodes, but they are well tolerated in mammalian cells. The identified compounds constitute a new class of broad-spectrum anthelmintics that could potentially be used in combination with existing drugs to overcome or delay the development of drug resistance. Another project we are pursuing is the identification of novel Bacillus thuringiensis crytoxins as potential biocontrol agents to tackle the nematode infection and the red palm weevil pest. Bacillus thuringiensis, or BT, produces crytoxins that are harmful to insect larvae but harmless to human cells. We are conducting a BT screen to find new biopesticides, as BT is a major biopesticide used in organic farming, and we're conducting the screen against the red palm weevils. The red palm weevil is a pest that bores into palm trees and is causing significant damage to date palms, a major crop in the UAE. Our goal is to identify new toxins that could be used to tackle this pest. In addition to our collaboration with our New York lab, we have engaged in several collaborative activity with labs here at NYU Abu Dhabi and in local university, UAEU, and with international institutions such as the University of Glasgow, St. Joseph University in Beirut, and University of Utrecht.
Hi, my name is Xinxue. I'm working as a research scientist in CGSB. Our research project in CGSB is related to finding novel bioactive compounds from natural resources. The natural resources we are interested in is marine bacteria, because the harsh environment in Abu Dhabi region hosts a unique community of marine bacteria which are unexploited. We are very interested in finding novel compounds from this collection of bacteria isolated in Abu Dhabi regions with the potential therapeutic use. Up to now, our lab have collected more than 200 pure bacterial strains in Abu Dhabi regions. From these collections, we now established a pipeline to fraction and extract pure compound from these bacterial cultures. Our efforts are now trying to identify the compounds with activity against parasitic worms, pathogenic bacterial infections, as well as bioactivities in mammalian cell models. We have identified several bioactive compounds target candidates. We are now studying their chemical natures and the mode of actions.
The main technology we're using to characterize bioactivity is a high-content screening system. It is an automated image acquisition and analysis platform that can be used to profile the cellular phenotype after genetic or chemical perturbation. We have established a staining panel in mammalian cells to capture the cytologic features in response to chemical perturbations. We are now planning to profile a library of compounds with known mechanisms and use their profiles as references to predict the mode of action of novel compounds.
We are also building a novel bacterial room dedicated for pathogen bacteria. In this room, we plan to screen for novel compounds targeted on antibiotic-resistant pathogens. Our high-content screening platform also contributed to the research in NYUAD. We have established several collaborative projects with established faculties in NYUAD. And we believe our platform will continue to contribute to the ongoing research projects which involve high-content image analysis.